Biochemistry, 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 biochemistry.
Hello everyone. We from Group 7. In this video, I will explain about cells and macromolecules, namely macromolecules. The topics that will be discussed are proteins, nucleic acids, polysaccharides, and lipids.
The first thing is protein. Protein is a polymer of amino acids linked together with peptide bonds. Amino acids can exist in the form of pairs of D and L stereoisomers, but only the L isomer is found in protein. There are 20 types of amino acids which are dipolar ions or sweaters, ions in water solution and can be acidic and basic, so they are called amphoteric. The picture shows the general structure of an amino acid which is differentiated based on its R group. Proteins have both structural and functional roles.
The second is nucleic acid. Nucleic acid consists of DNA and RNA. Nucleic acid is a nucleotide polymer, meaning it is composed of many nucleotides. Each nucleotide is composed of nitrogen bases, pentose sugar, and phosphoric acid. In short, it is phosphate sugar. There are three main types of RNA in cells, namely messenger RNA or mRNA, ribosomal RNA or rRNA, and transfer RNA or tRNA. All three are found in both prokaryotes and eukaryotes.
Nucleic acids play a role in the storage and processing of genetic information, but the expression of this information requires proteins.
Next, polysaccharides. Polysaccharides are polymers of simple sugars that are covalently bound with glycosidic bonds. Their main function is to store sugar, and as structural materials, cellulose and starch are components that are abundant in plants. Both are polymers of glucose but differ in the way they are monomers. Glucose is linked. Cellulose is the main structural component in plant cell walls. Starch functions for sugar storage and is found in a large number of intracellular granules or within cells, which can be hydrolyzed quickly to free glucose for metabolism.
Cellulose is a linear polymer with one greater than 4 bonds, about 40 parallel chains forming horizontal seeds or sheets that stack vertically one on top of the other. The chains and sheets or Z are tied together with hydrogen bonds to produce fibers that are strong and insoluble in water.
Starch contains two components, namely amylose, a linear polymer with 4 bonds, and amylopectin, a branched polymer with additional 6 bonds with a maximum of 106 glucose residues. Amylopectin is the largest molecule known. The differences in bonds in starch produce a curved conformation that cannot be packed tightly so that the starch dissolves in water.
Tissues in fungi and some animals, for example liver and muscle, store glucose as glycogen, which is a branched polymer such as amylopectin.
Chitin is similar to cellulose, but the monomer unit is an acetylglycosamine.
Mucopolysaccharides or glycosaminoglycans form a gel-like solution in which connective tissue fiber proteins are embedded or inserted.
Determining the structure of large polysaccharides is complicated because they vary in size and composition, and because they cannot be studied genetically like nucleic acids and proteins.
Next are lipids. Individual lipids are not all macromolecules. Many individual lipids are composed of smaller monomeric units, and they are involved in many groups of macromolecules. Large lipid molecules are dominated by hydrocarbons as their constituents and are very insoluble in water.
Some of these lipids are involved in energy storage and transport, while others are key components of protective membranes and other cell structures.
Glycerides have 12 or 3 long chains of fatty acids which are attached by ester bonds to the glycerol molecule. In animal triglycerides, the fatty acids do not have double or saturated bonds, so the chains are linear or straight. These molecules can be packed tightly and produce solid fats. Plant oils contain unsaturated fatty acids with 1 or more double bonds. The angular structure of these chains prevents fixed packing, so they tend to be liquid at room temperature.
Membranes contain phospholipids, which consist of glycerol esterified to both fatty acids and phosphoric acid. Phosphate is usually also esterified to small molecules such as serine, ethanolamine, inositol, or choline.
Membranes also contain sphingolipids such as ceramides with a long chain of the amino alcohol. Sphingosine has a fatty acid linked by an amide bond that attaches phosphocolins to the ceramides to produce sphingomyelin.
Then you